Hi, my name is Bernard, and I would like to talk to you about ApiNATOMY. ApiNATOMY is a multi-center effort that brings together expertise in computer science, image processing, bioengineering, and medicine to manage knowledge in physiology and pathology. In this particular presentation, I would like to overview briefly the ApiNATOMY rationale of building topological maps of tissue communication routes across anatomical scales. Now, in general, physiology studies biophysical operations across multiple spatial and temporal scales. These operations are typically represented in terms of the transfer of energy from one form to another or from one anatomical location to another. Computational physiology, for instance, has achieved significant progress in developing mathematical approaches that describe and quantify biophysical operations in terms of these energetic transitions. In so doing, this field has generated an impressive array of models that simulate these events. However, this effort has also given rise to considerable fragmentation and duplication of effort within this field of modeling. This shortcoming is partly due to either poverty or, it has to be said, incoherence in the explicit representation of multiscale anatomical location within these models, or indeed their associated data. In practice, the explicit representation of anatomical location associated with physiology models is either too poorly described to begin with, or when anatomy is described, it is depicted by non-standard means. This shortcoming has led to an inadequate search capability of community-shared modeling resources, that is, giving rise to duplication of effort, of course, as relevant resources cannot be found, as well as a lack of interoperability when resources are indeed found. This, of course, leads to fragmentation as models cannot talk to one another, and this, of course, discourages reuse. Now, given the above obstacle, the goal for the ApiNATOMY effort is to produce a biophysically informed dissection of standardized multiscale anatomy and to coherently link physiology models and data via the results of this dissection. The emerging technique of ApiNATOMY circuitboarding provides the computational means to achieve this linkage through the use of standard ontologies that represent anatomical components from different scales. As computer-readable knowledge graphs of terms and their relations, these ontologies of biological structure provide standardized information about the parts and about the subtypes of anatomical components across multiple scales. At this point, we'll focus briefly on the biophysical rationale underlying the ApiNATOMY dissection and how the standardized and formalized representation of multiscale anatomy may support integration, interoperability, and visualization in biomedicine. In this presentation, we particularly focus on the application of the ApiNATOMY approach in molecular physiology and pathology, and here's why. In previous work, we have argued that the regulation of molecular interaction is a key function of tissues, such that the organization of our knowledge of tissue structure in support of physiology studies should be informed by these functional characteristics. To link tissue structure with function, therefore, we must focus on the role tissues play in both short and long-range fluid transport necessary for the co-location and interaction of molecules across the body. On this basis, a key goal for ApiNATOMY is to create a detailed map of fluid transport that will act as a scaffold over which physiology models from different spatial scales may communicate effectively with one another. This approach resulted in the development of a multiscale route map of fluid transport for the body, which we depict graphically as a circuit board. In particular, the representation of a route requires the explicit depiction of flow modality and location of flow. In its current implementation, the ApiNATOMY circuit board represents explicitly two modalities of fluid flow, namely advective and diffusive. The advective modality takes into account the long-distance fluid transit along either multicellular tubes lined by endothelial, epithelial, mesothelial, or other cellular films like synovial membrane, pia mater, as well as unicellular tubes such as neuronal axons and dendrites, or Philippodia in osteocytes. The diffusive modality, of course, takes into account fluid transit across cellular compartments. For location of flow, we take into account where flow is taking place. This knowledge is explicitly represented in terms of community-supported standard reference ontologies at organ, cellular, and subcellular level. Before we view some tangible examples of ApiNATOMY circuit boards, it may be useful to point out that, by convention, the circuit board display adopts the following graphical rules. 1. Flow processes are displayed as edges. And 2. Tiles depict anatomical location, with nesting of tiles describing parthood and subclass relations over a tree map derived from ontologies of biological structure. The ApiNATOMY toolkit per se provides the means to navigate tree maps, the topology of which is derived from standard reference ontologies such as the foundational model of anatomy or mouse anatomy. The layout of an anatomy tree map, such as the one we observe here, is constrained to allow the relative spatial location of tiles to convey particular meaning. For instance, the whole-body tree map here is constrained to create a continuous epithelial border on the perimeter or coast of the map. The landlocked tiles in the center make provision for advective cabling to be laid down. For example, here we create a circuit board with edges representing advective flow of blood overlaid onto the relevant location tree map of tiles. The ApiNATOMY toolkit provides a graphical user interface for navigation and interaction with the elementary components of a circuit board. A second circuit board example of flow process knowledge is that derived from the neuroscience information framework, showing neural tracts traced in the central nervous system via various experimental approaches. ApiNATOMY currently supports the searching and interaction with circuits of either neural or cardiovascular tracts, or both. The metadata repository underlying the ApiNATOMY front end enables the visual interaction with different kinds of biomedical resource. 3 types of resource already interoperating with the circuit board include molecular expression and interaction data. Seen here are protein expression and interaction data from the brain. These proteins are also associated with small molecule interaction data, obtained via the OpenFacts service, as well as domain architecture data available via the Ensemble resource at the European Bioinformatics Institute. Other resources include geometric models from different scales, such as volumetric representations of organs from BodyMaps 3D, as well as reconstructions of neural cells from neuromorpho.org, as well as mathematical modeling environments such as OpenCMIS from the Auckland Bioengineering Institute. A key feature of the ApiNATOMY circuit board is the integration from inception of knowledge about primary functional tissue units, known as PFTUs. PFTUs are generated from 3-dimensional histology reconstructions of small advective vessels, such as capillaries or nephrons, and from these reconstructions we calculate the diffusive field that surrounds them. Primary functional tissue unit derived geometric data, therefore, provides the boundaries for a mathematically well-defined field of transition between advective and diffusive flow modalities. We are currently also analyzing cellular architecture patterns within PFTU fields to capture geometric signatures of cellular layout in support of both automated tissue classification, as well as spatial modeling of molecular events across these cells. Our long-term goal is to provide a public knowledge base of richly annotated primary functional tissue unit data for normal tissues, and to continue to embed and link this knowledge onto the circuit board. The inclusion of explicit location information into the ApiNATOMY map has two key implications. The first is that physiology models and data that are annotated with standard reference ontologies may now be automatically connected with one another over the root map. A second, more long-term implication is that clinicians and mathematical modelers may now collaborate to combine expertise via the circuit board. The ApiNATOMY circuit board in general, and the flow routes in particular, make use of terms from standard reference ontologies as building blocks. A key goal for the ApiNATOMY effort is the application of these ontologies to imbue model elements such as variables or parameters with precise meaning in terms of anatomical location. This application of annotation provides the means to automatically infer the physiological relationship between models or their corresponding data over location parthood, subclass, or topology of flow root. In practice, ApiNATOMY also provides tools for annotation and automated inferencing. These tools further support and incentivize the annotation of data and model resources by the community. Overall, annotation with standard reference ontologies adds value to resources through semantic linkage with other sources of knowledge that share the same semantic standards. ApiNATOMY is also being developed to provide the means for disease experts to describe multi-site and multi-scale pathology mechanisms in terms of the location and process knowledge depicted on the circuit board. Given the automated inferencing functionality discussed previously, mathematical modeling of pathology processes would be directly guided by the contributed knowledge about disease mechanisms, and would also benefit from the curated topological root data, as well as semantic interoperability with public physiology resource repositories. It will be possible, for instance, to bring to bear peer-reviewed models on the study of complex disease processes. In that sense, the ApiNATOMY approach provides a potential avenue to manage complex knowledge about anatomy in direct support of multi-scale modeling. Our goal is to further enhance the computational physiology effort through the provision of richness and coherence of anatomy location knowledge to match the mathematical rigor and eloquence that already typifies the repertoire of physiology models developed by this community. Thank you.